In this video, we will discuss about how the T cell activity works against these tumor cells. So, in our previous videos, we have discussed various other aspects of this T cell act against tumor cells, such as checkpoint inhibitor therapy, and other is the CAR T cell therapy. In the checkpoint inhibitor therapy, we will we will try to inhibit the checkpoints that we will try to inhibit or block the checkpoints that inhibit or that disrupts the T cell functions to act against tumor cells. And in the case of CAR T cell therapy, we artificially introduced the receptors that are present on the tumor cells. And in this case, the T cells that is used is V gamma 9V delta 2 T cells, and this kind of T cells can detect these receptors that are present on the tumor cells, that is BN2A1, BTN2A1, BTN3A2, BTN3A1, and the receptors on the T cells that detects or that interacts with this this BTN receptors on the tumor cells are this V delta 2 and V gamma 9. So what this research recently published research has find out is that there is a signaling pathway changes, or there are multiple changes occurred in the tumor cells that leads to the expression of this kind of receptors on the tumor cell surface. So one of the significant finding that they have done is the disruption in this because tumor cells are under stress, that leads to the upregulation of this signaling pathway, that is AMPK. And what are the stresses that leads to the upregulation of this pathway is this we can say oxphosis or glycosylations, purine, and biosynthesis. So disruption in all this will lead to this AMPK signaling pathway, and simultaneously the hypoxia condition, which is very predominant in the tumor cells, also promote this AMPK upregulation. And what happens in this AMPK upregulation is that there is a post-translation modification happens, and within this there is a change in the post-translation modification happens, and that leads to the formation of this kind of BTN kind of cell surface on the BTN kind of receptor on the cell surface of these tumor cells. And other factors that are that plays a role in expressing this BTN kind of receptors is this end-linked glycosylation and silation. And these studies has find out that there is a also changes in this silation and end-linked glycosylation occurs. End-linked glycosylation pathway has been observed in this study, and that is in line with the upregulation of this kind of receptors on the cell surface. And in terms of this AMPK signaling pathway, what they have find out is that what they have find out is that this, because of the stresses the tumor cells are under, that might lead to the upregulation of this signaling pathway, that is BTN32A, 3A1, and 2A1. And the pathways that inhibit the upregulation of these receptors are been inhibited or are been downregulated or inhibited, such as these ZNF217, CTB1, RUNX1. All these are inhibited, and in the case of tumor cells, whereas in case of normal cells they should be activated so that these kind of receptors are not formed in the normal cells. And apart from this IRF1 and NLRC5 and these pathways promote the upregulation of these kind of receptors are also upregulated in the found in this study in the case of tumor cells. So there are several factors that lead to the upregulation of this kind of pathway. And previously what people are finding out, what people are attempting, or even the clinical trial is attempting, is to increase the amount of T cells to detect to detect more tumor cells with this BTN2A1 receptors. However, the novelty of this study is that they have found out the uh, intracellular changes that occur in the tumor cells that can lead to this kind of receptors, so that now we can target on this. Now someone can target or on one of these factors to further upregulate this kind of receptors, and they are more, so that they can be more predominantly detected by the this kind of T cells, that is V gamma 9V delta 2 T cells. And the immune response of these T cells will finally leads to the death of the tumor cells within the patients. So I hope you have understood this new finding of this T cell activity on tumor cells, especially of this V gamma 9V delta 2 T cells. If you need to understand more about these details, you can go through the reference Nature paper that has been provided in the description. That's all for this video. Thank you for your time.